Okay, so my name is Zuhaib Ali Khan. I'm doing PhD at University College London under the supervision of Dr. Paul Hillier and Professor Nekos Ladomatis. The topic of my presentation is effect of temperature and residence time on the pyrolysis of methane and ethane in a high temperature flow reactor. Methane and ethane are the important component of natural gas, while the methane is the main constituent of biogas and from anaerobic digestion and is used in transportation and industrial power plant. The question is how these, The question is how these aliphatic hydrocarbon end up in forming benzene and polyaromatic hydrocarbon, which has environmental and health impact. To understand this, the following objectives were set for this research. First is to study the influence of increasing carbon chain length and residence time and temperature on the intermediate species. Second is the possible pathway that leads to the first ring formation, that is the benzene. And the third is the onward growth of the benzene to the pHs via acetylene or other intermediate species. Motivation: air pollution has reached critical level in many major cities, endangering public health, deteriorating environment, and causing property and landscape harm. The particulate emission, which contribute to air pollution, vary greatly in size and composition and able to penetrate deep into the human lung, conveying carcinogenic polyaromatic hydrocarbon present on the particle surface. However, despite continuous strict emission regulation necessitate to further examine the decomposition pathway of the hydrocarbon fuel and investigate the potential precursor to the formation of the benzene ring and the pHs which contribute to the air pollution. This figure shows the schematic diagram of the tube reactor, indicated a gaseous fuel supply line and a sampling pathway for intermediate sample collection. In all experiment, oxygen-free nitrogen was used as a carrier gas with a fixed flow rate of 20 liters per minute. Gaseous fuel were injected in the vaporizer using a mass flow controller. The pyrolysis of the methane and the ethane were carried out within a temperature range of 869 to 1213 degree at a fixed carbon rate of 10,000 ppm. The sample temperature at the inlet of the probe were measured. The sample temperature reported here is corrected for furnace radiation effect. Intermediate gaseous species were collected at different residence time with the help of specially designed high temperature ceramic sampling probe. The sampling probe was connected with a vacuum pump and a rotameter. Gaseous samples were collected via glass meter through the septum. After collection, the sample were immediately injected into the FID for further analysis. This is the high temperature sampling probe. So a sampling probe was specially designed for the collection of intermediate fuel pyrolysis species, which were injected into the furnace and held at a specific depth to dilute and freeze the reaction immediately. After the sample has been withdrawn, a flow of nitrogen was supplied and metered via mass flow controller. Air was used to cool the sampling probe. The temperature of the sample probe and inlet of the sample were recorded. 10 intermediate species up to C6, which contribute to the formation of the benzene ring and the pHs growth, were studied in this work, focusing in particular on the possible pathway for the formation of the benzene ring. These 10 species are shown here. Methane and ethane were chosen because of their simplicity, which allowed the observation of the structural changes, that is increasing carbon chain length, as it is important fundamental property of all carbon-based fuel. The sample were collected at 5 residence time and a range of temperature as shown in this table, but due to time constraint, only a few residence time and temperature will be discussed here. Methane pyrolysis. So this figure shows the pyrolytic breakdown of the methane at the longest selected temperature residence time at different set temperature. It can be observed from this figure that methane is very stable up to 1018 degree and even at 1120 degree, only half of the methane is pyrolyzed to other species. However, when the temperature was increased to 1213 degree, the conversion of methane was exponential, suggesting a higher pyrolytic rate. So with the methane fuel, it is necessary to reach a temperature above 1018 to produce gaseous species like ethylene, acetylene, ethane, and benzene. And for methane, a temperature above 1018 is presented here for further discussion under its pyrolytic breakdown. This figure 5 shows the effect of residence time on methane pyrolysis. Figure 5a shows that at the shortest residence time, that is 747, there was no intermediate species, and the only component detected by the GC was the fuel, suggested that the fuel breakdown has not yet started. But as the residence time increases, the concentration of intermediate species also increases. It can be seen that at a residence time of 1120, more ethylene is produced as compared to acetylene, but as the residence time increases, acetylene overcomes the ethylene reaction. For example, at a residence time of 11866, the concentration of acetylene is almost twice that of ethylene. It is interesting to note that as the concentration of acetylene increases, the concentration of benzene increases as well, which suggested some sort of equilibrium. This might also suggest that the formation of the benzene is via the cyclotrimerization of acetylene as reported in literature. This figure 5b shows that at the highest tested temperature, the pyrolytic breakdown of the methane test fuel started at very lowest residence time with the highest concentration of all intermediate species. And as the residence time increases, the concentration decreases, suggested the possible conversion of these species to higher ring pHs, species like naphthalene or higher, and finally to the soot mass. It is also interesting, it is also suggested that at higher temperature and higher residence time, the conversion of acetylene to benzene and the growth of the pH is very fast as compared to lower temperature and residence time. This figure 6 shows the effect of different gaseous species at constant residence time and to set temperature. As mentioned earlier, a temperature excess of 1018 is required to observe other gaseous species. Therefore, no species were detected at this temperature other than the fuel at all residence time. Figure 6a shows that as the temperature increases, the concentration of acetylene, ethane, and benzene increases, while the concentration of ethylene decreases. It can be observed from this figure that the ratio of acetylene to ethylene increase with increase in temperature. At a temperature range of 1018 to 1120, ethane and benzene were not detected. It is suggested that to see ethane and benzene at lower residence time, a temperature higher than 1120 is needed. This figure B actually shows that everything peaks at the medium temperature, that is 1120, and higher temperature, majority of these species has passes to higher species and possible to soot. The higher concentration of acetylene, at higher temperature as compared to ethylene suggested its role in the formation of the benzene and the pH's growth via the Haka mechanism. This figure 7, this shows the effect of residence time on ethane pyrolysis at two different sample inlet temperature. Figure 7a shows that the main product of the ethane pyrolysis were ethylene, acetylene, followed by methane. It can be observed from this figure 7a that at lower temperature, more and more fuel is converted into ethylene and acetylene. It is also interesting to note that once methane is formed, its concentration remained constant at all residence time at this lower temperature. This observation confirmed the finding from the methane pyrolysis at this lower temperature about its stability. At higher concentration of ethylene, at this lower temperature regime suggested that its role in promoting molecular weight along with acetylene. The concentration of acetylene increased with increase in residence time. This figure 7a also shows that acetylene was found in relatively higher abundance, while a small concentration of the benzene was detected by the FID, suggested that the benzene form quickly converted into higher species via the addition of acetylene to the Hecke mechanism. It can be observed that as the residence time increases, the relative concentration of ethylene increases, while the relative concentration of acetylene decreases, suggested a possible conversion of this double bond ethylene to acetylene. This figure 7b shows that the major product detected at this temperature are acetylene, followed by ethylene and methane. It can be observed from this figure that acetylene peak with no fuel left. It might be interesting to note that either the heavier species break down again and form acetylene, but as we only look for the species up to C6, so these higher species were not detected. Next is this figure 8, which basically shows the effect of temperature on the pyrolysis of the ethane at two constant residence time. It can be observed from this figure 8 that the amount of ethane in the mixture quickly reduces as the temperature rises, being substituted by ethylene. The concentration of ethylene raises before it reaches about 1018. At this point, there is no substantial amount of ethane present, suggested that the dehydrogenation of ethylene to acetylene become the primary reaction. At the lowest residence time, benzene was not detected and the concentration of the methane decreased with increase in temperature as expected. This figure 8b shows the effect of temperature on ethane pyrolysis at the longest tested residence time. And it can be seen that at the lowest temperature, ethylene is mostly converted into methane, ethylene, and acetylene. While at higher temperature, the absence of all these species suggests that they have converted into higher species higher than benzene. Conclusion. So the detection of intermediate species during pyrolysis shows that the cooled sampling probe successfully freezes the reaction. Second, increasing the carbon chain length affected the initial breakdown of the methane as methane needed higher initiation temperature, and the relative abundance of intermediate species, that is C1 and C2, and interestingly, C3 and C4 species were not detected. Third, acetylene was a common element in the pyrolysis of these tested alkene hydrocarbon, which would serve as a precursor to both surface growth via the Hecke mechanism and the pH's formation via cyclization. Fourth, the relative abundance of C2 species highlighted the abundance of the Hecke mechanism in the case of these fuel. The distribution and the toxicity of the pHs formed by the Hecke mechanism from methane and ethane pyrolysis will have health impacts. In the future, we will analyze butane and some oxygenated fuel and we will compare them. Thank you so much.